For most of human history, infections left no written trace. There were no doctors, no diagnoses, and no medical records. Only the bones remained. But now, those silent witnesses are speaking again through ancient DNA. In a groundbreaking international study led by evolutionary geneticist Eske Willerslev from the University of Copenhagen and the University of Cambridge, scientists extracted ancient DNA from the teeth and bones of 1,313 individuals ranging from Ice Age hunter-gatherers in Siberia to medieval farmers in Europe. The study, published in the journal Nature, mapped a timeline of human infections stretching back 37,000 years. Inside these remains, they found the genetic traces of 214 different pathogens, including viruses like hepatitis B and bacteria like Yersinia pestis, the cause of plague. These were not rare occurrences. Nearly 3/4 of the individuals studied carried some kind of infection. In the ancient world, illness wasn't an exception, it was a constant companion. The earliest infections were mostly oral bacteria, still common in humans today. But a dramatic shift occurs around 8,000 years ago, when humans began farming, storing food, and living in close quarters with livestock. By 6,500 years ago, DNA evidence shows the consistent appearance of zoonotic pathogens, those that jump from animals to humans. This includes early strains of plague, as well as cowpox relatives, leptospirosis, and meat-borne yersiniosis. These infections didn't just threaten survival, they shaped evolution. The study revealed changes in our immune system genes over time, especially those linked to inflammatory responses. Many of these genes helped ancient people survive deadly infections from animals. But today, they're also linked to autoimmune disorders, such as Crohn's disease or rheumatoid arthritis. The very genes that once protected us can now misfire in cleaner, industrial environments. Some skeletons even carried multiple infections. One Viking-era skull showed traces of both hepatitis B and plague DNA. Co-infections like these may have caused sudden death, and may explain rapid mortality events seen in ancient graveyards. Interestingly, the study found no clear genetic evidence of tuberculosis. Not because it wasn't present, but because TB bacteria often stay in the lungs, and don't leave as much DNA in the blood or bones unless the person was actively sick at death. This ancient DNA dataset doesn't just reveal the past, it helps prepare for the future. By comparing ancient and modern pathogen genomes, researchers can identify stable protein regions, the kind vaccine makers rely on to target microbes. For instance, older strains of plague lacked the YMT gene, which modern plague bacteria need to survive in fleas. That mutation marked the transition from local outbreaks to a global pandemic threat. Ultimately, this research shows that our immune system wasn't shaped in hospitals, it was forged in contact with animals, in early villages, in pandemics long before history began. The story of disease is also the story of us. And our DNA remembers.